Thank you very much for including me in this exciting event. It's wonderful to be here. I was thinking as I was sitting through the talks that we're having a bit of a therapy session. It's like a group therapy session. We're all in recovery from the century of the gene, where you had this prime mover that controlled everything that flowed from it, right? And now we get to think about multitudes and layers of relationship and those kinds of things. So I'm going to do more of the same, and talk about many of the things that have come up in talks already today, and turn us not inside out, but outside in. So I did biology in the early 1990s, and it was a time of the ascendance of the Human Genome Project, and the central dogma was very certain, and the sort of order of things was very clear to people. Today I spend a lot of time roaming around in the life sciences in an ethnographic practice. And I've come to think of it as a time of ontological fracture. So our categories that might have seemed quite clear, gene and environment, insides and outsides, senses and viscera, are under enormous pressure. They're under enormous pressure from idea change, the emergence of the microbiome, under enormous pressure from many of the problems in the world that our explanatory frameworks just aren't adequate to answer. So things like antibiotic resistance are putting enormous pressure on classical ideas of what microbes are and how simple they are and all of those things, or metabolic disorder, for example. So perhaps because fracking is a big presence in the world, I'm thinking about fracturing, this enormous pressure on a landscape of categories. And what happens is not that the categories dissolve or go away, but they do break into pieces, and they start to enter into new relations with one another. So what I want to do with my talk today, which is about microbiomes and senses and smell and those kinds of things, is to practice some ontological fracturing with these categories by telling you 3 stories. And you'll see that they come together in the end. Okay, so my first story is about smells and blood. And I use a paper by one of my colleagues at UCLA, Utpal Banerjee, who studies fruit flies. And he found, along with colleagues, that if you were to get fruit flies to lay eggs in food that was stripped of odor, the egg would develop into a larvae, but things went wrong in the development of the larvae in the absence of odor. What happened was that you have an odor, and the odor, if you had food which had odor, this was what would happen. You would have an odor, it would meet olfactory receptors that were specific to those odors, which would in turn inside the developing larva excite brain neurosecretory cells, which would make GABA, a neurotransmitter. And this GABA would act on stem cells in the developing larvae to develop a progenitor pool for fly blood. You might never have thought of flies as having blood, but they do indeed. So in the presence of odor, you get this normal developmental trajectory, right? And what they found in this experiment was if you put a fly larva in food that is completely adequate in terms of its nutritional content, its energy content, but it simply has no smell, the developing larva does not make an intact blood system. It doesn't develop properly. So in this way, we see that the presence of an environment through smell is essential to the normal development of the organism in this case. So in other words, to put it really simply, no smell, no fly, right? It's an example of the environment being constitutive of the developing organism. So this maybe shouldn't surprise us, and of course any sane fruit fly would not lay an egg in something that didn't smell, because smell tells the fruit fly that that's a good place to put its eggs, because when they hatch there'll be something to eat. Because where does the smell come from? The smell comes from microbial fermentation of fruit sugars and things like that larvae like to live on. So there's a fascinating field, actually, of the study of odor as semiochemicals, or informative signals between organisms. And here you have some scientists saying that insects are especially sensitive to odors because they advertise nutrient sources, habitats, or they're aversive. They advertise predators. But these scientists observe that even though the power of odor has been studied and has been known to influence animal behavior, it's only recently that we've come to realize how much of that is, to use a term from earlier today, in symbiosis with microbes. The way that animals use their relationship to microbial populations to shape their behavior and to seek each other out and those kinds of things. So that's the story of smells and blood in flies. My second story is about noses and kidneys. Those are not two things that you might have thought to put together, but we will put them together. Here is a nose, or more specifically a nasal cavity. What this textbook diagram shows you is the olfactory epithelium, this lining of the nasal cavity. And in this olfactory epithelium is where we have found the basis of smell, the human power of smell. You have these receptors that are specific to particular odorants. So in the technical terminology of a receptor, that is just the right shape for a particular ligand. And that ligand, the smell of broccoli, whatever you like, is specific to that receptor. So in the molecular era, smell receptors as physical things were tied back to a particular gene family. They're called G-protein linked receptors. And because you can look for the pattern in the genome of that receptor, you can find the receptors now through just finding them in the genome. And discovering, of course, that their role goes well beyond just nasal smelling, that after they were discovered in the early 1990s by Linda Buck and Richard Axel, they were also found to be present in sperm and to function in chemotaxis. So this sort of idea of there being a ligand specific to a receptor is not just about flies finding food, but about all kinds of entities finding each other. The ability to identify these receptors meant that you could find them everywhere, but you didn't necessarily know what they were specific to. And in a lovely terminology, these receptors where they didn't know what they were specific to were called orphans. They were orphans because it was not known which smell or which particular chemical ligand they were specific to. So if we fast forward to about 2014, I'm going to tell you the story of some orphan receptors that were found on the surface of kidney cells. So these G protein coupled receptors were, much to the surprise of investigators, found in kidney cells. And now they were surprised to find them there because they were thinking of them as smell receptors. But of course, they're called smell receptors because we found them in the nose, not because they are intrinsic, they don't have smell written on them or anything like that, right? That's the term that we have given to them. And then when we find them somewhere that's not the nose, it's a surprise. So from this paper by Jennifer Plusnick, who's the lead investigator on this work, they write that G protein-coupled receptors play key roles in organs and tissues traditionally thought of as non-sensory. We don't think of the kidney as a sensory organ, for example. Indeed, Jennifer Plusnick tells the story of thinking that she had pulled the wrong sample out of the freezer when finding these smell receptors in the kidney, because what would smell receptors be doing there? And it was a orphan receptor. It was not known what the ligand was that was found. So it's expressed in nerves and smooth muscle cells in the kidney. And one of the interesting things about this orphan receptor was that when it was stimulated, it had this physiological effect of kicking off a cascade of signals inside the cell that released renin, which is a hormone which drives blood pressure up. So it seemed to be linked to the physiology of blood pressure. Now, here's a picture of the urine flowing by these cells in the kidney that these receptors were on the surface of. And perhaps you can guess where I'm going, that when it was figured out what the ligand for these cells were, it became clear what the connection was between needing this kind of chemical sensing in the kidney. So short-chain fatty acids, which came up once before, are these, they're called short-chain because they have short chains of carbons. Acetic acid, propionic acid, and butyric acid. This is just the name of the salt of the acid, acetate, propionate, butyrate. And these orphan receptors were parented, I guess you would say. How do you become unorphaned, adopted? By finding that they were specific to acetic acid and propionic acid. Where do these things come from? Why are they flowing through the body? Why are they interesting to kidney cells? Well, they're metabolites. They are produced by bacteria in the colon in the process of digesting carbohydrates. So humans eat a lot of things that they actually possess no ability to digest, strangely enough. Human cells have no enzymes for breaking down cellulose. Human enzymes have no ability to break down things like oat bran. You may be familiar with these substances because they're called prebiotics in today's food fashions, right? They are things that feed bacteria. So when humans eat these so-called non-digestible long-chain carbohydrates, the bacteria in their colon eat them. They digest them and they produce these short-chain fatty acids. This is bacterial poo in a way, right? It's the product of bacterial metabolism. So in the colon, these substances are produced in high volumes, especially after a meal. Butyrate tends to stay more local than the other two, and the human cells lining the colon, their primary energy source is butyrate. So most cells in the body use glucose, but the ones around the colon use butyrate. These other two, in larger volumes, cross over into the hepatic portal vein and get carried through the bloodstream. And it turns out that they are these specific ligands for these receptors in the kidney. So the short-chain fatty acids are released during bacterial fermentation in the colon. They're transported into blood, and they find these receptors which are specific to these very molecules made only by bacterial metabolism. When the receptor is triggered, it triggers human cells to release renin, which drives up blood pressure, which circulates the blood faster, which moves nutrients around the body. Right? So it makes sense now, right? You've found the ligand for the receptor, and you see that there is this mechanism by which the colon is signaling to the kidney to change physiology in order to facilitate digestion. So here's just an image of the kinds of papers that you find this in. An olfactory receptor responding to gut microbiota-derived signals plays a role in renin secretion and blood pressure regulation. So here's just a picture of the short-chain fatty acids moving through the bloodstream, meeting the receptors, and then the renin being released and changing that physiology. As if that wasn't complicated enough, they also found a second receptor, GPR41, which has a slightly different binding affinity for short-chain fatty acids. So after renin release is set up and blood pressure goes up, after a while, these ligands attach to this receptor and blood pressure goes back down again. So bacterial digestion is basically driving blood pressure in this situation. Now, it was surprising to Plesnik to find smell receptors in the kidney, but since then, a whole festival of discovery of what are now called chemoreceptors has occurred. We find bitter taste receptors on the surface of the lung. We find receptors that detect pH in the spinal column. These are part of sour taste receptors in the tongue. Even better, a synthetic sandalwood odorant is found to stimulate wound healing in the skin, again mediated by these kind of chemoreceptors. So a smell is driving a cellular change. Okay, so that's the second story. The third story is about butyrate or butyric acid. Here's the structure of butyric acid. So the smell of butter, or better, the smell of rancid butter, can be attributed to butyric acid. And now you know why there would be a lot of butyric acid in butter. It is a microbial metabolite. Cows have lots and lots of bacteria in their rumen, and they're busy metabolizing. Butyric acid was actually discovered by the chemist Michel Chevreul in the early 20th century. And it is, as I said, short-chain fatty acid produced by bacteria in the colon. It accounts for 3 to 4% of the composition of butter. It is responsible for the distinctive smell of Parmesan cheese, kombucha, and also vomit, which may explain to those of you who don't like kombucha why you don't like kombucha. It can be detected at 10 parts per billion by dogs, who are very good at smelling things, and 10 parts per million by humans. It's a very strong smell. And again, it is a ligand for these receptors, not just in the nose, but throughout the viscera, throughout the internal tissues, adipose tissues, lymph nodes, bone marrow, and seems to play a very important role in things like appetite and satiety. The global market value of butyric acid is estimated to be $124 million in 2015 and projected to grow by about 15.1% between 2015 and 2020 to this size in that year. Why on earth, right? Why would we need so much of the smell of butter? That is a very good question. I've told you about the smell and its operation on the insides at these micro scales. So why are we mass manufacturing it? Well, the key driver turns out to be the animal feed market. Again, you might say, well, why? Well, much of the food that our food eats is not the pasture outside of the farm building, but comes to them in a feed bag. And butyric acid, being the smell of butter, makes the feed appetizing to them. It also plays a role, as it does in humans, in protecting intestinal integrity. It's something that's very important to industrial agriculture. I'll also add that butyric acid is very important in the chemistry of making flavors. So if anybody has ever opened a bag of Cheetos next to you, you know where some of this butyric acid is going. Butyric acid participates in an economy of what flavor and odor scientists call phantom aromas. We associate the smell of butyric acid with the taste of butter or cheese. Therefore, we can have foods that have butyric acid signaling to us that we're about to eat cheese, and we feel like we're eating cheese, but there's no cheese there. So this is another role of butyric acid in our world today. Some of the butyric acid in this market is made by bacterial fermentation because obviously they're good at it, right? So you can put a lot of bacteria in a vat and make a lot of butyric acid. But according to industry sources, low production and high costs are factors limiting the growth of renewable butyric acid. Also, the pungent smell of butyric acid limits its direct use in applications. So most of it actually comes from petrochemicals. We crack petrochemicals, much like the process of fracking, much like the process of ontological fracture, to break it into these component parts and make things like butyric acid from it. So the majority of this smell in the world is manufactured from petrochemicals. So what do you get if you add the three stories that I just told you together? Let me take you through it. We get from the first story that the inside, not just of fruit flies, but anything that develops, is constituted by the world, right? The outside becomes inside. We also get the feeling now that sense is not just a feature of the receiving surface. It's not just the olfactory epithelium, it's not just the eye, it's not just things on the surface that sense things and then go on to do their own operations, right? Sensing is a visceral property. So things that we might have thought of as smells or tastes are physiologically, developmentally, and mentally operative well beyond the senses. And then we get to the final part, right? The industrial scale capture of these processes, the industrial scale capture of microbial metabolites, and the synthetic production of semiochemicals from petrochemicals is remaking the world. And if the inside is constituted by the world, then this is a cycle, right? And we are remaking worlds in which organisms are developing and being. So it's important not just to understand the biology of these lovely objects that we work with in the laboratory, the fruit flies, the embryos, the bacteria. They're really hopeful stories. It's really a wonderful kind of turn in biology. It makes everybody feel kind of good in a world of not so good stories. But I'm afraid that I'm re-embedding it for you in the fact that we also have to think about these things in the context of the world that we have made through industrialization. So I'm going to end by proposing that we don't just marvel at these stories of new kinds of biologies, but to think about how to live with them and how to live better with them. So I've been thinking about the way in bioethics that we have genetic ethics. Should we engineer our babies? We have neuroethics. Should we, use human neurons to make little brains in the dish? These kinds of things. But perhaps what we really need is a metabolic ethics. So a metabolic ethics is a framework for viewing human action in relation to human biology that foregrounds a scientific and philosophical understanding of life as a metabolic process. So you may note that I haven't really focused on the human cell or the bacterial cell, but the protagonist for my story has been the short-chain fatty acid. The short-chain fatty acid is the process of sensing and relating, right? So if we think of any object in the world as only ever coming into being through these chemical relations, then we start to see life as a metabolic process. This is another way to put it from Mary Poppins. And after all, he went on, flicking his terrible little forked tongue in and out as he spoke, it may be that to eat and to be eaten are the same thing in the end. My wisdom tells me it is probably so. So this is part of the process of becoming outside in, right? To eat and to be eaten. Our bacteria, ourselves, to eat and to be eaten. So I'm not alone in this. The philosopher of biology, John Dupre, can you make the movie go by any chance? Has said that what's stable and robust in biology are not things, but processes. And this is hard to think about because we have, again, I said we're in recovery from the gene, right? These very, very static objects. So to think of any body, any life, any being as a process, it's a little hard to think about it as not being a thing. But it is one way to get beyond the sort of endless productivism of the 20th century, where the body was a factory and a productive factory. What if the process is the end, the good life, the end of ethics, right? Not the end of ethics, but the purpose of ethics. What does it mean to have a good process? Not how is this process best at producing something profitable or good, but what does it mean to live a good process, to have a good process? Very simply, in the story that I've told you, one thing is we probably shouldn't be taking all the fiber out of our food if it's so constitutive to the process that is being both bacteria and human. So that's just an example of how this abstract thing could be a very practical thing. So I also like it because it's a kind of endless end. You don't ever stop. Life never stops being a metabolic process. So that is the target of metabolic ethics. So that's the end. Thank you.